Welcome to the BioInsights, where science comes alive.
Every protein in your body begins with a surprisingly small group of molecules, amino acids. What makes one protein an enzyme, another an antibody, and another a structural fiber? The answer begins with just 20 standard amino acids.
Every protein in your body begins with amino acids, the fundamental building blocks of proteins.
Every amino acid contains a central carbon, an amino group, a carboxyl group, hydrogen, and an R group. The central carbon is called the alpha carbon, and its three-dimensional arrangement is extremely important.
The variable R group gives each amino acid its unique chemical and physical properties.
R groups determine polarity, charge, interactions, and ultimately influence protein structure and function. Amino acids join through peptide bonds, releasing water and forming longer polypeptide chains.
Polypeptide chains fold into precise three-dimensional structures that allow proteins to function.
Amino acids can be classified according to the polarity and charge of their side chains. Non-polar amino acids have hydrophobic side chains and commonly avoid contact with water.
During protein folding, hydrophobic residues often move toward the protein interior, away from water. Phenylalanine, tyrosine, and tryptophan contain aromatic ring systems with important molecular interactions.
Polar amino acids interact favorably with water through hydrogen bonding and other polar interactions.
Aspartate and glutamate are acidic amino acids with negatively charged side chains at physiological pH. Lysine, arginine, and histidine are basic amino acids containing proton-accepting nitrogen groups. When amino acids enter water, their ionization state changes according to the surrounding pH.
In water, amino acids commonly exist as zwitterions, carrying both positive and negative charges.
A zwitterion contains opposite charges, but can have an overall net charge of 0.
The charge of an amino acid changes as the surrounding pH changes. At low pH, amino acids gain protons and become more positively charged. At high pH, amino acids lose protons and become progressively more negatively charged.
Protonation is reversible, creating an equilibrium between protonated and deprotonated forms.
The pKa tells us how readily an ionizable group gives up its proton.
When pH equals pKa, the protonated and deprotonated forms are present in approximately equal amounts.
Below its pKa, the protonated form generally predominates. Above its pKa, the deprotonated form becomes increasingly dominant.
The Henderson-Hasselbalch equation connects pH, pKa, and the ratio of protonated to deprotonated forms.
As pH rises, the balance shifts toward the deprotonated form.
Titration allows us to observe how an amino acid gains or loses protons as base is gradually added. As base is added, the measured pH generates a characteristic amino acid titration curve. Near a pKa, the amino acid system resists changes in pH and acts as an effective buffer.
Glycine provides a simple example because its amino and carboxyl groups are both ionizable.
At low pH, glycine carries a positive net charge because both groups are protonated. At intermediate pH, glycine mainly exists as a zwitterion with 0 net charge.
At high pH, glycine loses another proton and carries a negative net charge. The isoelectric point, or pi, is the pH at which an amino acid has 0 net charge. For amino acids without ionizable side chains, the pi lies between the two relevant pKa values.
Most amino acids are chiral because their alpha carbon has 4 different groups attached. These mirror image forms are called enantiomers and have different 3-dimensional arrangements. Ribosomes use L-amino acids to build proteins in living organisms. Glycine is different because its side chain is hydrogen, making it achiral.
Biochemists use standardized 3-letter abbreviations to represent amino acids efficiently.
Even shorter 1-letter symbols allow entire protein sequences to be represented rapidly.
The order of amino acids in a protein is called its primary structure. Interactions among amino acid side chains help the chain fold into its functional 3-dimensional structure. Hydrophobic residues often become buried inside proteins, while many polar residues remain exposed to water.
In membrane proteins, hydrophobic residues often interact with the membrane's nonpolar interior.
A concept map connects these ideas and makes the relationships between amino acid properties much easier to remember.
For exams, focus on structure, classification, charge, pKa, titration curves, pi, and chirality. Tiny changes in amino acid chemistry can produce enormous differences in protein structure and biological function.
Understand amino acids, and you begin to understand the molecular language of life. This is the BioInsights.
Thanks for watching the BioInsights. Like, subscribe, and follow for more.